Assalamu alaikum class. We are going to start a new topic. We will discuss bacteria. First of all, we will discuss some important things about bacteria, and then we will go for classification. So, classification of bacteria is quite lengthy. So, don't worry. But classification is also easy. So, let's start our today's lecture. We will discuss what are bacteria and why bacteria are so important for human beings. As many type of bacteria are present inside our body, but why we are getting effect from bacteria means most of and there are a huge number of infections are caused by bacteria in human body. So, we will discuss the role of bacteria in human disease, and then we will go for classification. And most importantly, you have to keep in mind the basis of classification because bacteria are classified on number of bases. So please keep in mind how many types of how many categories of bacteria have. So let's introduce bacteria. Bacteria are microscopic single-celled organisms that thrive in diverse environment. Okay, these organisms can live in soil, the ocean, and inside the human gut. Human's relationship with bacteria is complex. First of all, you have to keep in mind bacteria is a unicellular organism, as we have already studied in our previous lecture, which was that was about the micro classification of microorganisms. So bacteria are unicellular organisms and are prokaryotic. Okay, so they are mostly there are huge number of bacteria present in soil and human gastrointestinal tract is also contain many huge number of bacteria uh, uh, and and in many diseases, and there are huge number of diseases for humans and for animals as well are caused by bacteria. So there is a complex relationship with bacteria and human because there are some friendly bacteria and there are some pathogenic bacteria. So both type of bacteria are present in a same condition. So that's why we can say bacterial relationship with human is actually a complex. Sometimes bacteria lend us a helping hand, such as by turning milk into yogurts or helping with our digestion. Uh, as there are many bacteria are present in our GIT tract, so they help in digestion of food. Okay, or some bacteria produces vitamins like vitamin K, vitamin B, and in our intestine. So they are also beneficial for us. And in our industrial scale, bacteria's used for yogurt formation. It converts milk into yogurts, and they also there are number of applications our industry due to with the help of bacteria. So actually bacteria is a unicellular microscopic organism which have some important which have friendly as well as pathogenic relationship with human, and they also help human at industrial level as well. Here you can observe some examples of bacteria, how bacteria appears in different shape. There are some cocci, then bacilli, and there are some with spore formation. So it is actually just a diagram to show you how it appears. So classification of bacteria, they are actually we can classify bacteria on two major classes on the basis of phenotype of bacteria. It means actually appearance of bacteria. It means some characteristics of bacteria, and we can classify on the basis of genotype of bacteria. But we will mostly concentrate on phenotype, because classification, most important classification of bacteria is on the basis of morphology, culture characteristics, motility, etc. But please keep in mind phenotype जो है please keep in mind phenotypic classification is much important. And now uh, uh, we are going to start phenotypic classifications of bacteria. So let's arrange some phenotypic categories of bacteria. If we classify bacteria on the basis of physical or phenotypic characteristics, and we can say we can classify bacteria on the basis of morphology, on the basis of their anatomical structure, on the basis of their staining reaction. And then we can classify on the basis of cultural characteristics, nutritional requirements, and how environmental factors can affect bacteria, and on the basis of biochemical reactions, and then antigenic structure. So there are there are many, there is a list of categories of phenotypes. So so let's start. They're most important. So let's start morphological classification of bacteria. So on the basis of morphology, or on the basis of appearance of bacteria, we can classify bacteria into four categories. Cocci, those bacteria which appears round in shape. Bacilli, those bacteria which appears usually exist in rod shaped, or in the form of bacilli. And then another type is spirilla. A spirilla means spirochetes is an example of spirilla. They exist in the spiral form. They exist actually. They are different from cocci and bacilli. Then last one is Vibrio. Vibrio is actually a comma-shaped bacteria. They exist, or their normal shape is comma shape. So in cocci, some bacteria can exist in a single cell, just with a single cell. They are actually a monococcus. So if a bacteria can exist single, or cocci bacteria can exist single, then we can get a monococcus. And sometimes bacteria exist in the form of pair. Cocci bacteria uh, exist in the form of pair. So these cocci bacteria, those cocci bacteria which exist in the form of pair, are known as diplococci. Diplococci, you can see in diagram. So diplococci bacteria, most common example of diplococci bacteria is enterococcus. Okay, some bacteria exist as some cocci bacteria or coccus bacteria exist as in the form of clusters, because their plane of division is in all fashion. So they exist in the form of clusters, or like we can say grape like structures. Okay, then we can say it's actually a staphylococcus bacteria. Sometimes cocci bacteria exist in the form of chain. They survive in the form of, they forms long chain instead of clusters. Then we can say it's a streptococcus bacteria. Okay, so it's a streptococcus bacteria. Their anatomy, their morphological shape is round, but their arrangement is in the form of chain. Then if we move in the for bacillus, there bacillus is also consists of streptobacillus, diplobacillus, etc. Spirilla, a common example of spirilla is spirochete. Vibrio, a common example for vibrio is vibrio cholera bacteria. Okay, we can classify on bacteria on the basis of their anatomical features, like we can arrange bacteria on the basis of anatomical structure in capsule, in capsule, flagella, spores types. It means we can bacteria can have different bacteria may have capsules, or sometimes they don't have capsule. On the other hand, if some bacteria have flagella, other don't have flagella, and some bacteria can produce spores and other cannot produce spores. So let's take a review of these anatomical features of bacteria. So what is the role of capsule? Last time in UHS there was a question, write down the role of capsule in bacteria. So don't worry, we will discuss capsule in bacterial organelles. When we study the organelles of bacteria, then we will see capsule in detail. Now, how we can classify bacteria on the basis of capsule? So, some bacteria are capsulated. There is a polysaccharide. There is a special type of capsule present around the outer membrane. This is known as this is known as actually a capsule, and those bacteria which contain a capsule or special structure outside the outer membrane. These are actually capsulated bacteria. Not every bacteria is capsulated. Common example is Streptococcus pneumoniae is a capsulated bacteria, and another type is non-capsulated. Those bacteria which exist without capsule, they survive without capsule. They don't have any genetic information to create capsule around it. So they are actually non-capsulated. Viridin streptococci are the common examples. So there are many types of examples for capsulated, and there are many types for non-capsulated bacteria. So let's move forward. We can also classify bacteria on the presence of flagella on the basis of flagella. If flagella is present, then we can say bacteria is flagellate, and if flagella is not present, then we can say bacteria is aflagellate. Okay, so or sometimes we can say it's a atricus. Okay, aflagellate or atricus, both are same actually. So Shigella species are bacteria responsible for dysentery in human is a Shigella. So Shigella is atricus or aflagellate. Monotricous bacteria in flagellates. Monotricous bacteria are those bacteria which have a single flagella at one end. They are actually monotricous. And if bacteria have tuft of flagella, bunch of flagella at one pole, then they are lophotrichous. If bacteria have flagella on both sides, means on both poles or terminals, then we can say they are amphitrichous. And there are some bacteria which have a lot of flagella on whole surface. Okay, so they are known as peritrichous bacteria. So flagellates bacteria are divided into 4 to 5 categories: monotrichous with single flagella at one end, lophotrichous with bunch of flagella at one end, amphitrichous means flagella at both ends of bacteria, and the peritrichous means a lot of flagella present in whole surface of bacteria, not only both ends but whole surface. So they are actually peritrichous. So this was the classification on the basis of the presence of flagella. In fungus, usually fungi spread through spores. They can survive in harsh conditions with the presence or with the formation of spores. But now there are few bacteria which can which can survive with the spore formation. They are spores are actually a resistant thing or a resistant species in microbiological world. So bacteria there are some bacteria, they are spore forming, they can survive, survive for a long period of time in the form of spores. So they are spore forming. They are actually a bacillus species. For example, bacillus species are common spore forming bacteria commonly present in soil and environment. Then another thing is non-spore forming bacteria. Non-spore forming bacteria, those bacteria which cannot they don't have actually any genetic makeup to produce spores. They are unable to produce spores. So common example is Escherichia coli. So another thing is classification of bacteria is we can classify on bacteria on the basis of their staining reaction. First of all, we can say the gram positive bacteria, and secondly we can say gram negative bacteria. Third, we can say acid fast bacteria. Actually, in our microbiology laboratories, there are two types of stains commonly used. First one is gram stain, and the second one is Zn strain, Zil Nelson strain, Zn strain. So let's talk about first two, gram positive and gram negative. Some bacteria, please keep in mind, some bacteria have thick peptidoglycan layer in cell wall. Some bacteria have thick peptidoglycan layer in cell wall, so they can stain with a crystal violet and appears usually blue in color. Okay, so in gram staining reaction, gram positive bacteria are actually those bacteria which have thick peptidoglycan layer in cell wall, and this thick peptidoglycan layer can bind with the crystal violet, and at the end it appears these bacteria appears blue in color. So they are actually gram positive bacteria. And what are gram negative bacteria? Those bacteria which have not thick peptidoglycan layer in cell wall. In gram negative bacteria, cell wall is composed of with lipopolysaccharides instead of peptidoglycans. Okay, it means in gram negative bacteria, lipopolysaccharide is present in huge number. It cannot stain with the crystal violet, and usually appears red in gram staining reaction, red or pink in gram staining reaction. So they are actually gram negative bacteria. So on the basis of gram staining reaction, we can classify gram positive bacteria and gram negative bacteria. Gram positive bacteria are those bacteria which have a thick layer of peptidoglycan, okay, in cell wall. And gram negative bacteria are those bacteria which are actually lipopolysaccharide instead of peptidoglycan. So crystal violet is actually a reagent or is a dye which binds with the peptidoglycan. So if peptidoglycan is there, then crystal violet can strongly bind, and gram positive appears blue in color. But in gram negative, lipopolysaccharide is present instead of peptidoglycan. So safranin, or you can say secondary dye, binds with the membrane cell wall instead of crystal violet, because crystal violet binds with the peptidoglycan. So if peptidoglycan is not present in the gram negative bacteria, then safranin binds with the lipopolysaccharides, or safranin gets binds with the bacteria. So we can say they are actually gram negative. They appear usually red or pink in color in gram staining reaction. So on the basis of gram staining reaction, we can say there are two classification: gram positive bacteria, and those are gram negative bacteria. And third category is acid fast bacilli. This acid fast bacilli, what are first of all, you can say the common example is acid fast bacilli, Mycobacterium tuberculosis (TB). Mycobacterium tuberculosis, a bacteria. Uh, Mycobacterium tuberculosis or acid fast bacilli have thick mycolic acid layer. Okay, a thick mycolic layer is present outside of the cell wall, or at the boundary of cell wall. It means bacteria is protected with the thick mycolic acid layer. So those bacteria which have mycolic acid in cell wall cannot be stained with the gram stain. They need so for the diagnosis, or to visualize acid fast bacilli, we need special type of stain, and that is Zn stain, Zell-Nelson stain. And the bacteria appears usually red to pink in color on the basis of with the Zn stain reaction. And another type is wall-less bacteria. They don't have any cell wall, like mycoplasma. So they are we are unable to stain with gram staining or Zn staining. So that's why we can say that these are wall-less bacteria, and common wall-less bacteria is mycoplasma. Common example is mycoplasma. Mycoplasma is wall-less bacteria. Here we can see we can classify bacteria on the basis of their cultural characteristics. What is culture? What is culture? We have studied the artificial. Uh, we can grow, or we can obtain a bacteria in artificial medium for the diagnosis, for the study, for the research. So this mechanism is yani कि artificial growth is known as culture. So we can there are bacteria appears different on different culture medias. Some bacteria need special requirements of growth factors. Okay, special requirements. They are extra growth factors requirement. Some bacteria show hemolysis on blood agar. Okay, and some bacteria shows utilize carbohydrates, and they usually utilize carbohydrates in other two manners. And some bacteria are slow grower or fast grower. It means they have different growth rate. And some bacterias, not all bacterias, but some bacterias can produce a color, different color, okay, on different culture media. So these are actually cultural characteristics. We can classify bacteria on the basis of cultural characteristics. So if we classify bacteria, if we study bacteria, those bacteria which need extra growth factor requirement, we can classify fastidious we can classify into fastidious and non-fastidious bacteria. Fastidious are actually those bacteria which need actually a special requirement. They need extra growth factors. But non-fastidious bacteria, they do not need any special requirement for growth factors for survival of bacteria. So Neisseria species, Neisseria can cause meningitis. Neisseria can cause gonorrhea in female. So Neisseria is a fastidious organism, and the non-fastidious organisms are common, just like Klebsiella, Escherichia coli. They are non-fastidious. They don't need a special food requirement. Another thing is hemolysis. Another base of classification was hemolysis on blood agar. You can observe in diagram beta hemolysis, alpha hemolysis, and gamma hemolysis. Some bacteria show beta hemolysis on blood agar. It means a complete hemolysis of red blood cells in the culture plate. So some bacteria show partial, incomplete hemolysis of red blood cell in a culture plate, and some bacteria shows no hemolysis at all in a culture plate. So this is actually a gamma hemolysis. So beta hemolysis is a complete hemolysis, alpha hemolysis is a incomplete hemolysis, and gamma hemolysis is no hemolysis at all. So there are many bacteria which show these phenomena. We will discuss in our next lectures. Bacteria can utilize carbohydrate with two fashions. First of all, they can oxidize a carbohydrate. For example, micrococcus utilized the carbohydrate by oxidizing them, and some bacteria can ferment carbohydrates, and they can convert into special type of product. So these common fermentative bacteria is Escherichia coli, but both are utilizing carbohydrates, but in different fashion. Another thing is classification is bacteria on the basis of growth rate. Some bacteria can grow rapidly, like Vibrio cholera. It can move. It can grow rapidly in a culture plate or in any medium, if it have a proper nutrients available. As there are another thing is slow grower. Those organisms which have slow doubling time. They have low doubling time, like Mycobacterium tuberculosis. Okay, so bacteria can we can classify bacteria on the basis of growth rate as well. Some are rapid grower and some are slow grower. Not every bacteria can produce pigment or color product. A few bacteria can produce pigments, like Pseudomonas aeruginosa can produce five types of pigments, like blue, red to brown. They can also produce yellow color. Some bacteria can produce pigment, and some bacteria cannot produce pigment. They are actually non-pigment producers. Common example is Escherichia coli. Common example of non-pigment producer. Oh, there are huge numbers of non-pigment producer, but pigment producers are Pseudomonas. Serratia marstans can produce Serratia. Marstans can produce red color on nutrient agar. Okay, so they are pigment producers. They have a genetic makeup to produce a color or a pigment on culture plate. Okay, so on the basis of pigment production, we can classify yes or no category. Those pigment producers, those without pigment production. And another classification based on nutrition, bacteria usually or commonly get their food from hosts. They are autotrophic. They are not autotrophic. Common bacteria are not autotrophic. They are heterotrophs. Like in human body, many pathogens are heterotrophs. Many pathogenic bacteria are heterotrophs, and some bacteria, like most of them, are present in green environment, or common in water, like cyanobacteria. They are bacteria, they're single and unicellular organism, but they can produce their food. They are autotrophs, like cyanobacteria are autotrophic organism. Okay, so based on class nutrition, we can classify bacteria into autotrophs and heterotrophs, and commonly no, human pathogenic bacteria as heterotrophs. Bacteria need a special requirements to survive according to environment, because some bacteria can survive in high temperature or high pH, but many other bacteria cannot. Similarly, we can classify bacteria on the basis of environmental factors. What types of environmental factors? Like temperature, oxygen requirement, pH, salt concentration in environment, and atmospheric pressure. We classify bacteria on the basis of temperature. Then we can say psychrophiles. Psychrophiles are those bacteria which can survive within 15 to 20 degrees centigrade temperature. They are usually a moderate temperature, 15 to 20 degrees centigrade temperature. They are actually, we can say, sometimes in some books you will learn psychrophiles are 4 to 20 degrees, survivors within 4 to 20 degrees centigrade. Okay, but common is 15 to 20 degrees centigrade, like Pseudomonas fluorescens. Another type is mesophiles. Mesophile can survive within 20 to 40 degrees centigrade. Now we can say common human pathogenic bacteria are mesophile, because they can cause disease at 37 degrees centigrade. Okay, so Escherichia coli, Salmonella, Staphylococcus aureus. Thermophiles are those bacteria which can survive at uh, more than 50 degrees centigrade or 50 to 60 degrees centigrade. It means they can survive in they can survive in high temperature environment. So a common example is Bacillus stereothermophilus. This bacteria is also used for the monitoring of autoclave. Okay. So, another condition is extremely thermophile, those bacteria which can survive up to 250 degrees centigrade. Those are extremely thermophile bacteria, but are mostly rare in environment because our environment is around is have a temperature around 20 to 45 or 50. On the basis of oxygen requirements, we can say bacteria can be aerobes. Those what are aerobic bacteria? Those bacteria can grow in which can grow in the presence of oxygen. Okay, they are aerobic bacteria, obligate aerobes, they need a strict requirement of oxygen. Okay, aerobes bacteria are those bacteria which actually can survive in the presence of oxygen, but obligates aerobes are those bacteria which are only oxygen dependent. They can die in the presence of carbon dioxide, or they can die in the presence of they can die in the absence of oxygen, or in the they cannot grow in insufficient oxygen. Okay, so they are obligate means strict oxygen require. Those bacteria which require strict oxygen supply. Microaerophilic are those bacteria which grow under reduced oxygen supply, usually 5 to 10% increase carbon dioxide. It means we can say microaerophilic are those bacteria which needs increase carbon dioxide like 8 to 10% carbon dioxide. They are Campylobacter jejuni, Helicobacter pylori. Another type is facultative anaerobes. Those bacteria which are actually anaerobics. They do not need oxygen. They can grow in the absence of oxygen. But what they are actually anaerobic. But what are facultative anaerobes? Facultative anaerobes are those bacteria which can even survive in the presence of oxygen. Okay, anaerobic are those bacteria which can die in the presence of oxygen, but facultative anaerobes can survive, can grow in the can grow in the aerobic condition as well, can grow in the presence of oxygen as well, but they facultative or anaerobic bacteria needs absence of oxygen. Another thing is obligate anaerobe, those bacteria which need a strict anaerobic environment. Oxygen presence can be harmful for those bacteria like Clostridium species. They cannot survive in the presence of oxygen. Capnophilic bacteria require increased concentration of carbon dioxide. Okay, microaerophilic and capnophilic are slightly different. Microaerophilics, those bacteria which can survive in the low amount of oxygen. Okay, low amount or less amount of oxygen, and capnophilic needs a special or high concentration of carbon dioxide. Okay, like Haemophilus influenzae, Neisseria gonorrhoeae, and those some are aerotolerant. They are actually they can tolerate environment. Okay, if they are anaerobics, then if they are anaerobic, they can tolerate the presence of oxygen as well. So they are aerotolerant means they can tolerate the environment. They can survive in different environments. Okay, on the basis of pH, we can divide those are acid loving, which usually grow as in the presence of oxygen, like Lactobacillus acidophilus, and those are which can survive in the high a value of pH like alkaliphiles. For common example is Vibrio, and neutralophilic. Neutralophilic are those bacteria which can survive in neutral environment or slightly alkaline environment, like within the pH of 7.2 to 7.6. Okay, so majority of medically important bacteria grow best at neutral or slightly alkaline reactions. They are unable to replicate or survive in acidic conditions. On the basis of salt concentration, some bacteria are salt loving. They survive in the salt environment. Salt environment, we can say many bacteria present in oceans like halophiles. They are halophiles. Those are salt loving. They can survive in salt concentration, high salt concentration. Some are non-halophilics. They cannot survive in the presence of high salt concentration. Okay. Now the end of the lecture is some ways of other ways of classification. We can also classify bacteria on the basis of their motility or non-motility. Some bacteria are motile and some are non-motile. Uh, Salmonella is highly motile organism. On the other hand, Klebsiella is non-motile. We can say some are human pathogenic, and most of the microorganisms present in GIT, they are not causing any disease. They are non-pathogenic. Some are resistant toward any antibiotic, and some are resistant to antibiotic. We can classify, and we can classify also bacteria. They can ferment lactose. Lactose is a carbohydrate or sugar. So lactose fermenter and non-lactose fermenter is actually a classification in sub classification in carbohydrate utilization. Okay, so classification of bacteria is much important. अगर आपको कभी यूएचएस में क्वेश्चन आ जाए 5 मार्क्स का कि क्लासीफाई करें बैक्टीरिया और उसने मेंशन ना किया हो किस बेस पे करें, तो सारी क्लासिफिकेशन आप मेंशन करेंगे बिकॉज इट्स अ 5 मार्क्स क्वेश्चन. थैंक यू सो मच.